Modern biotechnology. Human insulin, as we all know, is a very important product of the industrial biotechnology applications. That is because there are millions of people in India alone that are suffering from diabetes mellitus, and we all know that synthetic insulin, okay, that is produced large scale in industries, is very important for the treatment of diabetes. Okay, so it is also a product of transgenic Escherichia coli. Now, what is the meaning of the term transgenic? Transgenic means a combination. Now, what combination? Combination means it is consisting of a host genome plus the gene of interest. Okay, it is a transgenic Escherichia coli strain. Now, Escherichia coli, we all know, okay, is a bacterium. It is a gut bacterium and it has nothing to do with insulin synthesis in the human body whatsoever. Okay, so human insulin synthesis is not the job of an Escherichia coli bacteria. So how then did they manage to make the Escherichia coli strain produce insulin in the industries? So the answer to that lies in the fact that the genome of the Escherichia coli is manipulated, okay, and the genome of this particular bacteria is fixed with the genes that are responsible for the production of the insulin hormone in the human body. Therefore, the bacteria produces the large scale insulin, okay, in industries. Okay, the Escherichia coli strain that contains and expresses the gene for insulin. See, now it is very important, obviously, to make sure that the bacterium expresses the gene that has been given in it. Now, you can genetically manipulate any number of organisms you want, but it is also very important to make sure that the organism in itself is able to express that gene in order to get the desired product. Now, proteins produced by transgenes are called recombinant proteins. Now, recombinant, as the word itself suggests, okay, it is a combination, okay, which is produced by the genetic manipulation of organisms. The production technologies based on genetic engineering are termed as modern biotechnology. So, this involves manipulation, okay, of genomes. And it is as recent as the 1970s. So the next slide. There are two main techniques of modern biotechnology. Now we know that modern biotechnology is nothing but the genetic manipulation. So there are two techniques that are mainly used. Number one is the genetic engineering that we've been talking about all this while. It includes techniques to alter the nature of genetic material, okay, to introduce these into host organisms and thus change the phenotype of the host organism. So phenotype is nothing but the, okay, expressed character. Okay, so it is the genetic engineering that ensures that the phenotype of the organism that we are using to get the product of our desire, okay, is expressed by the organism. Then comes the chemical engineering part that involves maintenance of sterile microbial contamination free condition. Okay, so it is the job of the chemical engineering technology to make sure that the cultures that are produced, okay, are kept sterile and microbial contamination free. You do not want interference from, say, viruses or bacteria themselves or fungal. Okay, there are many risks of fungal infections involved in very culture rich media. Okay, now in chemical processes, to have growth of only the desired microorganisms. So as long as the media or the culture that you are having is sterile and it is contamination free, okay, you can get the microorganisms and the products of your desire. Okay, and biotechnological products such as antibiotics, vaccines, enzymes, etc. To the next slide. Now, genetic engineering, as we discussed, is based on two main discoveries, that is presence of plasmids in bacteria and restriction endonucleases. Now you may wonder, what has the presence of plasmid in bacteria have anything to do with genetic engineering? But first of all, it is imperative for us to know what plasmid means. Okay, plasmid is usually a circular DNA, double-stranded DNA that are produced by bacteria alone. Okay, in eukaryotes, that is bacteria, in prokaryotes, bacteria, you have a extranuclear, okay, double-stranded DNA, and that is called the plasmid. Now what do I mean by the term extranuclear? So we all know that this is the bacterium cell, okay, this is the genetic material of the bacterium. Okay, so now the genetic material of bacterium is the nuclear. Okay, now extranuclear means it is present apart from the genetic material of the bacterium. Now this bacterium here possesses a double-stranded extranuclear circular DNA. Now why does it have this DNA? It is because this plasmid contains the genes that are not present in the nuclear DNA of the bacterium. Now it is obvious, isn't it? If all the genes that are required by the bacteria are present in the nuclear DNA or the genomic DNA, why have a plasmid at all? Because plasmid has some special genes, okay, especially antibiotic resistant genes. Okay, so this is something that renders the plasmids useful, because these antibiotic resistant genes are very important. They play the role of something called the selectable markers, which we are going to talk about very soon. Okay, these characterize the products. These actually decide whether or not the recombination has been taken place. Okay, so the plasmids is both used as the recombinant agent and the delivery systems. So suppose you have one organism that you need to inculcate a particular genome into. Okay, so how are you going to do it? There are different physical methods of doing it, but it is the plasmid that carries the transgenic genome into the desired organism cells. Okay, now that was one, that was the presence of a plasmid. Then restriction endonucleases, we'll talk about this soon because it requires a little bit of explanation. Now, techniques of genetic engineering includes formation of recombinant DNA. Obviously, as I was telling you, we are going to manipulate the genome of an organism in order for it to produce the desired products, and use of gene cloning. Now what is cloning? Cloning is actually producing the same copies. Okay, clones are genetically similar. Okay, so we produce a transgenic organism, then we know that the transgenic property has been successfully done. Okay, it has been successfully introduced into the organism. Now it is the job of the biotechnologists to make sure that they are produced in large numbers, and that is why the technique of gene cloning and gene transfer. Now we have a recombinant plasmid. Okay, it has the gene of interest inserted into it, as I've already told you here. Now it is important for you to put this in the cell of the organism that you wish to produce it from. Okay, so suppose you want to produce it in a bigger cell. Okay, so this plasmid has to enter this cell. So gene transfer is important. So genetic engineering also includes formation of recombinant DNA, use of gene cloning and gene transfer techniques. Now there are three basic steps in creating transgenic organisms or GMOs. Now what is GMO? GMO is genetically modified organisms. So these are not natural organisms that are found in nature. We are going to take a natural organism and inculcate in it the properties that we want it to produce or have. Okay, so GMOs are nothing but genetically modified organisms, but there are three only basic steps that are need to be taken care of in order to produce a successful transgenic organism. Number one is identification of DNA with desirable genes. I was talking about the human insulin. Now, are you going to look for the human insulin gene in a plant? No, that is impossible. It is very imperative for us to identify the source of the gene that we are interested in. Okay, so if we are looking for a human insulin gene, we are going to have to sequence the human insulin gene and have the exact copy of that, okay, in order to be inculcated into an organism to produce a transgenic organism. So it is important for us to identify the gene first, the source of the gene first. Now then introduction of the identified DNA into host. See, it is as equally important as identifying the DNA, integrating it into the host DNA. Okay, now it is not enough that we know the genes for the human insulin exists in the human genome. It is not important for us to know that. It is also just to know that, okay, it is also important for us to take that sequence and put it in a host, because we need a large production of the human insulin. Okay, so it is very important to also introduce it into the host. After this, maintenance of introduced DNA, obviously, because if you are going to leave the organisms just like that, okay, the transgenes just like that, with whatever media you want to, they may not grow, or they may grow in contamination, and the products that will be produced will have more contamination. So there is also the expenses, okay, part that plays a role in genetic engineering, especially in industrial applications, because the large scale production also means there is large scale purification of products to make sure you only get the pure products. So to more the contamination, you'll have the lesser yield, okay, of the desired product, okay, which makes it even more complex, and you have to be very meticulous and careful while maintaining the introduced DNA into the organism and breeding it further. That is when you are carrying out the gene cloning processes. Now next, tools for recombinant DNA technology. We have various techniques and tools that are used for the production of recombinant organisms. Now the first tool is the restriction enzymes. This is perhaps the most important tool of all, okay, and they are called molecular scissors. Now let us before jumping into the concept of restriction enzymes or restriction endonucleases, we need to know the different kinds of nucleases. Now what are nucleases? Okay, nucleases are those enzymes that are capable of cleaving or cutting DNA, RNA. Okay, they are called nucleases. They are capable of cleaving nucleic acids. Now, out of which there are three types: exonucleases, endonucleases, and restriction endonucleases. Now, the difference between exonucleases and endonucleases is that exonucleases cut the DNA strand. We are more specifically interested in DNA in this case, therefore I am explaining with DNA. So exonucleases cut the DNA at the ends of a DNA molecule. Okay, it does not cut anywhere in between the DNA molecules. This is the double helix. Sorry about the structure, but assume this is a double helix. So now this double helix is being cut by the exonuclease only at the ends. Okay, now you may also very nicely observe another thing. These cut only single stranded DNA. Okay, now endonucleases, but the case with endonucleases is different. What the endonuclease does is that it creates nicks, or it cuts the DNA, again single stranded, in between the DNA molecules. It does not have to be at the end itself. Okay, these cut anywhere in between as well. That's why the term endonucleases. Now restriction endonucleases. Now the term restriction itself tells you that this is a highly specific endonucleases. So this endonuclease, which is able to cleave the DNA molecule, the double-stranded DNA molecules in between the strands as well, okay, are specific restriction. So specific to what? These restriction endonucleases or restriction enzymes produce the cuts or nicks only if there is a specific sequence of base pairs. Okay, suppose let us assume there is a base pair sequence of AGCT AGC. Okay, so 5' to 3'. So AGCT AGC. Okay, now we know of a restriction endonuclease that cleaves the DNA sequence here. Okay, now there is another sequence called ACTG ACT. So do you think the restriction endonuclease cleaves this? No, because the restriction endonuclease is not the restriction endonuclease, it is not going to cleave it because it is sequence specific. Okay, now there are three types of restriction endonucleases, types 1, 2 and 3. Now what is the thing with types of restriction endonucleases? See, the types of endonucleases here is based on where the DNA is being cut. Okay, so the type 1 restriction endonuclease does not cut double stranded DNA. It cuts only single stranded DNA. Okay, and it has 3 subunits. After all, it's an enzyme and it has 3 subunits. It needs ATP, it needs magnesium ions, it needs adenosyl thionate. Okay, it also needs all of this to be able to work. Now what is a type 2 restriction endonuclease? This is the more commonly used endonuclease in biotechnology. Okay, now why is this the most commonly used in biotechnology? Is because it can cleave double stranded DNA as well. Okay, and it requires only magnesium ions to function. Okay, now what are type 3? Again, type 3 can cleave only single stranded DNA. It has 2 subunits and it is the same thing with this. It needs ATP, magnesium ions and adenosyl thionate, okay, to be able to work properly. Okay, so these were the types of restriction endonucleases. So whenever I say endonuclease in context of biotechnology, we are only going to talk about the type 2 endonucleases. This does not require ATP for work, which makes it more advantageous for the organism to be able to use it for cutting the DNA. Now, the nomenclature of restriction endonucleases. This may not seem like a very important thing, but there have been questions in the past about this in the question paper. So I think it is important for us to pay attention to how to name an endonuclease. Now, first the first letter, okay, is the bacteria's genus. Now you need to understand one thing here. The restriction endonucleases are usually derived from bacteria only. Okay, because bacteria carry plasmids. Okay, bacteria are the only things that have plasmids. So these endonucleases, restriction endonucleases, are used for cleaving the plasmids themselves. That is why both the plasmid and the restriction endonucleases source are bacteria or bacterium. Okay, while we are naming an endonuclease, the restriction endonuclease, the first letter of the restriction endonuclease is the particular bacterium's genus. Okay, the next 2 letters is the bacterium's species, the last letter is the strain of the bacteria. Now we all know we have different strains of bacteria, so we are going to use them for naming the restriction endonuclease. And then the numerical is the order of the isolation of enzyme. Now it is usually in Roman numericals. Okay, so I'll give you an example. There is a restriction endonuclease called the EcoR1. Now, so what does this tell you? EcoR1. The first letter is the bacteria's genus, Escherichium. Okay, Escherichium. Right? So we are deriving the first letter of the endonuclease from the genus name Escherichium. Coli. Coli is the species name, of course. So we are going to take the first few letters of the species name and put it here. So eco, R. What is the R? As I told you, the last letter represents the strain here. So the strain of this happens to be RY13. This is the strain of the Escherichia coli bacteria, Escherichium coli bacteria that are used to produce this restriction endonuclease. Now, 1. What is 1 here? The numerical is the order of isolation of enzyme. So this was the first enzyme that was to be isolated from the Escherichia coli bacteria. I hope it's clear. Now moving on to the other enzymes that are used. Now what are the other enzymes used? Why do we need to use other enzymes? It is simple. Now there is a DNA and you have managed to nick it, okay, or cut it. So you now have DNA fragments. Now what are you going to do with these DNA fragments? What is our intention basically? It is our intention to integrate it into the genome, the plasmid, or into the host genome. Okay, so this part of my fragment will go here, let's say. So in order to do that, you need a lot of other enzymes. The first one being DNA ligases. Now, what is the meaning of the term ligation? Okay, ligation is to be able to stick. Okay, so these are called molecular glue, as opposed to the restriction endonucleases that are called molecular scissors. So DNA ligases are molecular glue. So as the name itself suggests, they are operational in integrating the host genome by making the ends of the DNA fragments stick to the host DNA. So sticking here means via phosphodiester bonds. Now phosphodiester bonds, we all know, are a part of the DNA phosphate backbone themselves. So you need the phosphodiester bonds in order to make sure that the DNA of your interest or the gene fragment of your interest is sitting there in the host integrated DNA. Okay, so it is carrying out this job by formation of phosphodiester bonds. It requires ATP, of course. Next, alkaline phosphatases. Now these are a very not everybody tells you the importance of alkaline phosphatases. Now it is important for the DNA strands to recognize the 5' to 3' direction sequence before going and sticking into the host genome. Okay, so what alkaline phosphatases do is that they make sure there is a phosphate, okay, at the 5' end, so that the phosphodiester bonds can easily form and make the DNA fragments stick together. So that is the job of alkaline phosphatases, and they also obviously require the ATP. Next is reverse transcriptases. Now reverse transcriptases are not compulsorily used everywhere. It is only in case of viruses, or sometimes even, yeah, basically retroviruses. Okay, so reverse transcriptase, they defy the central dogma of biology, which is DNA to mRNA, mRNA to what? mRNA to protein. Now we all know this is the central dogma of molecular biology, but in case of reverse transcriptases, RNA, okay, is the main how do I say it, template. Okay, from RNA you have DNA. In order to carry out the process of RNA to DNA conversion, you need a reverse transcriptases. Okay, and the viruses, usually the viruses that possess this particular enzyme are known as retroviruses. Okay, HIV is one such examples. HIV is a retrovirus. It may be helpful for you to know this bit of information. So reverse transcriptases are not compulsory everywhere in the biotechnology field. Next comes the DNA polymerases. Now what does this word polymerase tell you? So polymerase means multiple, right? Polymer is multiples. So DNA polymerase means it is for the production of a single stranded DNA to a double stranded DNA. Okay, so basically what we are doing is we are using the DNA polymerases in order to get our product of interest, and it helps in multiplying. So I have one transgenic organism, let's say. So without DNA polymerases in the cytosol of the organism or artificially provided DNA polymerases, there is a chance, no, there's not a chance, there is never a chance of the organism not dividing. Okay, there is no chance for the organism to multiply its genome in order to get the desired double stranded DNA, only those which can be expressed to give you the proteins. Okay, so DNA polymerase is also very important. DNA ligases, alkaline phosphatases, all of these enzymes, as I've told you, they have biological sources. Okay, DNA ligases are produced from some bacteria themselves. Alkaline phosphatases are usually produced from T4 phages. And reverse transcriptase from obviously retroviruses. Even DNA polymerases are from bacteria. Now coming to very important part of the biotechnology, that is cloning vectors. Cloning, so cloning here is multiplication. Okay, so clones have what? Clones are, they have the same genetic makeup, okay, as the parents. Okay, so those are clones. Now what are clones? What are vectors? I told you what clones are. Now vectors, what are vectors? Vectors are vehicles. What are vehicles? What do you mean by vehicles? Vehicles is something that will transport the cloned or the transgenic, maybe transgenic genome, the manipulated genome from the culture medium or the culture host to the desired cell or the desired organism. Okay, so without vectors, we have very less possibilities of getting a good yield. Okay, now there are two types of vectors, namely plasmid vectors, and you guys have different kinds of vectors, but let's discuss plasmid vectors first. Now the examples of plasmid vectors is PBR 322 and PUC18. Plasmid, you already know what plasmids are. So plasmids are those that have a selectable marker and they can replicate on their own their double-stranded DNA structures, as you can see in the picture here. Okay, and one of these examples is PBR 322 and PUC18. Okay, now the P here indicates plasmid, and BR in this case is the name of scientist that produced it. Okay, we do not know what the numerical derivative is. Even here the plasmid, P stands for plasmid, but UC stands for University of California, where this plasmid was first produced. This is probably the 18th plasmid that they produced, which is why it is called the PUC18. So this is the nomenclature of the plasmid, the vector, the plasmid vectors that are produced. Now I will come back to the picture later, because we also need to know some criterion for choosing a plasmid vector. Okay, now there are, as I was telling you, different kinds of vectors, one of which is bacteriophage vectors. Now what are bacteriophages? It is very interesting. We know humans and animals getting affected by viruses and bacteria. So bacteriophages one such virus that affect only bacteria. Okay, so you can imagine the size of the bacteriophages. They are that tiny. Okay, so bacteriophage vectors, because they are that tiny, we are going to use them as vectors because they can easily permeate into any bacterial cell. Okay, so the most important and the most common phage that we use is the T4 phage and the M13 phage vectors, because there is a reason they choose T4 phages and M13, because their genome has been completely sequenced, that's one, and two, we have sites that can be manipulated and put into in order to transport the transgene into the host organism. Okay, now we have other vectors apart from the bacteriophage vectors, BAC, YAC, cosmids. Now BAC is bacterial artificial chromosome. Okay, this is yeast artificial chromosome or YAC and cosmids. Now the thing about these BAC, YAC and cosmids is that they can take up very large DNA fragments. Okay, up to maybe say it can take up to more than 100 base pairs, which is a big size for ligating into integrating into a host genome. So these BAC, YAC do the job very nicely and they take up large fragments of DNA. Now there are features of cloning vectors. Now this is what I was talking about. We'll get back to the picture and I will maybe able to make you understand completely the features of the cloning vectors. Now cloning vectors have to have something called the origin of replication. You might remember from your molecular biology classes that origin of replication is the place where the DNA replication begins. Okay, without this there is no identification of the DNA sequence that need to be replicated. So in absence of an ORI or an origin of replication, that particular DNA cannot be replicated, or a single strand can't be made into a double strand unless you have an origin of replication. There is also a selectable marker. Now, what is the importance of a selectable marker? Suppose I integrate one bacterial genome, not bacterium, let's say human insulin itself, into a Escherichia coli strain that will produce the human insulin for me. Now, what guarantees do I have that my gene has been integrated properly into the host plasmid? Okay, now there is a gene sequence. Okay, selectable marker. I suppose have a sequence like this. Okay, now this sequence is tetracycline resistant. Okay, tetracycline is an antibiotic and we all know that the bacterium has a plasmid which carries the antibiotic resistant genes, okay, in order to provide resistance against antibiotics. Now this particular bacteria, let's assume, has a tetracycline resistance gene. Okay, so I take a genome, part of genome, and integrate it here. So this is the integrated genome, this is the new genome. Okay, so I start culturing it. Now what happens when I culture is that we know that the transgene has been produced with the experiment that the bacterium has to survive in a media of tetracycline. Now this is tetracycline. See, I know that the genome has been placed next to the tetracycline resistant gene. So if the bacteria is expressing this sequence of tetracycline resistance, it means that my transgene has been successful. Okay, so the tetracycline should not be able to affect your transgenes, whereas my integration has not been successful. Okay, let's say, okay, this tetracycline is also gone while I am trying to push the DNA fragment into this. Okay, so let's see. Now what happens when I introduce the genome? There is no tetracycline resistant gene or the resistant sequence. So in tetracycline the bacterium dies. Okay, so this is how a selectable marker is used. Next, recognition sites. Recognition sites for what? Recognition sites for restriction endonucleases. We need this obviously because we do not need random cleaving in DNA of DNA in multiple places. Now we have technology that can sequence the genome and suggest the suitable endonuclease that are to be used. So therefore it is imperative for us to make sure there are some recognition sites in the vector in order for us to integrate the DNA. It will be more detailed explanation in our crash course classes maybe. So this is just a demonstration of how biotechnology can be simplified. Now, processes of recombinant DNA technology. We so far have seen the tools that are used in the recombinant DNA technology. Now we are going to see the recombinant DNA technology processes. So it is important to isolate the genetic material, of course, identify and isolate it from the source, and cutting of DNA at specific locations using restriction endonucleases, obviously, and amplification of gene. So this is gene cloning, okay. Cloning using PCR. We all know what PCR means. It is polymerase chain reaction. So polymerase chain reaction or PCR, okay. It was given by a scientist, discovered, or I don't know, he invented the method maybe, by a scientist called Carey Mullis in as recent as 1975. Okay, so that is how recent PCR is, and it has revolutionized biotechnology. Okay, now PCR we are using, as the name suggests, polymerase chain reaction. Suppose we have one strand of DNA, only one strand, we can use the technique of polymerase chain reactions with some primers, with temperature control and a lot of other things to give me 10 strands of the same DNA. Okay, so we are multiplying the copies of the DNA present. So that is polymerase chain reaction, and it's a chain reaction. Production of one DNA strand will give rise to another DNA strand production, because there is bases that are added, and production of one single strand will give a complementary strand, making it a two-stranded DNA. So that is how the chain is continued until all the reaction material is exhausted. So polymerase chain reaction, it has been most helpful in field of biotechnology especially. So some applications of PCR include DNA fingerprinting. So we know DNA fingerprinting is mostly used in forensic sciences. So you probably have, say, a drop of human blood, okay, you found somewhere, and what the forensic department does, that it very carefully scrapes off the remains, or maybe use solutes to take it off, and isolate the minuscule amounts of DNA from whatever cells is present in that sample, okay, and then we are going to try to match it to a specific person. So if there is very less quantity of DNA, you will have to carry out a PCR in order to make it a substantially good sample for you to process. So DNA fingerprinting is one of the very fields that PCR is used. Now, detection of specific mutations or microorganisms. Now, specific mutations that are maybe of our interest can be detected using a PCR, and microorganisms as well. So we know the genetic composition of a particular organism, or we have the genetic sequence of a particular organism. So if that microorganism is present, we can check it. Maybe a patient's sputum sample is taken and a PCR is run in order to find out if there are TB organisms in that sputum. So that is how you can detect microorganisms as well. In parental diagnosis, obviously, because we can take a minuscule DNA amount and find out if whether or not the child or the offspring belongs to a particular parent. So it is mostly involved in legal issues. Now, these were some of the quick tools that quick recap of tools and techniques that are used for biotechnology. Now, what are bioreactors? Now, you may be wondering, there's so much technology used. So, what are we going to do it in? Is one culture plate enough to be used industrially? Of course not. So, that is where you use these huge vessels called bioreactors that can hold up to 100 to 1000 liters. Okay, we